Hey guys, welcome back to LabMate channel. Today we're going to talk about chocolate agar and what role it plays within microbiology section of the lab. So chocolate agar does not contain chocolate. Instead, it contains lysed red blood cells. That's why it has that distinct brown color. It's made just like blood agar, but instead the blood cells that are included into it are heated, and the cells then lyse, then releasing essential nutrients. So the lysed red blood cells release hemoglobin, X factor, which also is known as hemin, and V factor, which is also known as NAD, which are necessary for certain bacteria growth. Heating also deactivates V factor degrading enzymes, making the medium more supportive for growth. It supports the growth of organisms such as Neisseria and Haemophilus species. So in this table that we're looking at, we see the X and V factor requirements for Haemophilus species. So for Haemophilus influenzae, we see that both X and V factor are required factors for growth, while Haemophilus parainfluenzae only requires V factor, and Haemophilus raceae only requires X factor. Preparation of the agar. So when it comes to preparing chocolate agar, you start just like blood agar. So you prepare your base as per manufacturing instructions. Then you autoclave and sterilize. So you can use a base such as nutrient agar, for example, and follow the manufacturing instructions and autoclave and sterilize. Then you add blood, but instead of adding the blood at a lower temperature, you have to add the blood at a higher temperature because you need to lyse the blood cells. So after cooling the medium to around 80 degrees Celsius after it comes out of the autoclave, you add then 5 to 10% defibrillated sheep or horse blood, causing the red blood cells to lyse. You must gently mix the medium until it is homogeneous. Cool, and then pour into petri dishes and allow to solidify and store until use. So adding supplements. So supplements like batracin prevent overgrowth of non-pathogens, focusing on target organisms. So for Thayer-Martin agar, it is a modified chocolate agar used to isolate Neisseria species, and is modified by adding antibiotics to it. So quality control. So as per usual, you have your control plate, which is an uninoculated plate that is incubated to check for sterility. You want absolutely no growth on this plate. And then you also require a quality check organism. So for this medium, Haemophilus influenzae is a great one because it will show the growth. The growth will show that that the medium supports fastidious organisms to grow, and that also it will confirm the presence of both X and V factors, as per the table that we saw at the beginning of this presentation. The uses and limitations. So with uses, growth of and isolation of fastidious microorganisms as a differential media, routinely used in cerebral fluid cultures, antimicrobial susceptibility testing, and to perform the cytosol test to identify H. influenzae. The limitations. So non-pathogenic organisms can grow on this non-selective mediums as well, reducing specificity. But if Neisseria gonorrhoeae is suspected, Thayer-Martin agar can often be used because of its selectivity. So in the image that we're looking at, we see that there is a sample, a rectal specimen that has been tested on both chocolate medium and Thayer-Martin. We see that there's no growth on the Thayer-Martin medium, but we see that there's growth on the chocolate medium, which will mean that whatever organism has grown is not Neisseria species. Cool? Great. So when it comes to result interpretation, on chocolate agar, we're looking for distinct colony morphologies of fastidious pathogens. So for example, Haemophilus influenzae typically appears as small translucent colonies, as you can see in the image. You see these translucent microscopic looking colonies. Neisseria gonorrhoeae colonies are generally grayish, white, and shiny, while Actinomyces israeli colonies are white, rough, and elevated. So in conclusion, chocolate agar is essential for culturing fastidious organisms such as Haemophilus and Neisseria species. It's an enriched media, and its unique preparation releases nutrients that supports the growth of these challenging organisms. While it is widely used in clinical labs for diagnosing infections, especially from respiratory and cerebrospinal fluid samples. You guys can check out our references for further reading. Thank you guys so much for watching. Don't forget to like, comment, share, and subscribe. Hit us up and let us know that you guys enjoyed the video. You guys want more information? Do you guys want longer videos? Or do you guys want more shorts? What topics do you guys want us to discuss? Just hit us up. And thank you guys so much for watching the video in its entirety. Toodles!